Okay, welcome to this another Compass seminar. It gives me great pleasure to introduce today's speaker, Bernardi Bif, both because you will great science you will hear about in a few moments, and because Mari is a brand new faculty in our department, assistant professor in ocean sciences. Started in January 2nd, 3rd.
Yeah.
Right. So we are very lucky to have Mari joining us, accepting our offer. Just a quick background. Make my phone work. Right, Mari got her master's from Brazil in 2015, and then she moved to a PhD at this great school. Graduated with PhD in Professor Hansel's lab in 2019. And then she moved to California, working at MBARI in Southern California. That's right. At first as a postdoctoral associate, and then as a research scientist, doing research in biochemistry, working with biochemical data coming from those autonomous floats in the ocean, a real revolution in biochemical oceanography, I would say, as far as I know. Right, and Mari already is an author on 16 peer-reviewed publications, and she contributed to numerous talks and presentations, spent a few hundred days at sea on research cruises. So she already contributed a lot to science, and we're looking forward to her future contributions, and we're looking forward to her talk today. So please, stage is yours, and welcome to our department. Welcome to our school.
Thank you, Igor. Hi, everyone. I'm Mari from University of Miami now, previously at MBARI. I'm happy to see a lot of faces that I am not familiar with, a lot of early careers, because when I organized this talk, when I was thinking about what I was going to say, I didn't really want to talk about the in-depth science that I do, but I really wanted to introduce myself to you as a new faculty member, so you know who I am, what I do, what are the questions that I'm trying to answer, and also if you have anything that you feel like I could contribute to your research, that would be a nice way of showing how I can contribute, or how we can collaborate, or how you can think about using new methods and new data and different data to complement your work. So it's really the idea of this talk today. So from Rosenstiel School to Rosenstiel School is because I did full circle. I came here to do a PhD, and then I went out for 6 years, and then I came back, and I do biogeochemistry. So a little bit of what I'm gonna talk, a little bit of my story. How did I get here? Where did I went to school? And what did I study? Then a little bit about BGC-Argo floats. Maybe some of you don't know what BGC-Argo floats are. It's more or less a new technology that has been developed in the past 20 years, 15, 20 years. It's been collecting a lot of chemical data from the ocean. And it's making us, the chemists and the biogeochemists, looking at the ocean in a different way, using more large data sets that we were not used to do before. So I'm going to talk a little bit of BGC floats and their capabilities. Then I'm going to give you examples of 3 very recent researches that I have been conducting, just to showcase what are the cool things that we can do with BGC floats. And they're all about extreme events and changes in the ocean. So looking at changes in the ocean chemically and biogeochemically using these floats. And then I'm gonna touch, and then I'm gonna touch a little bit about future projects, what I'm gonna do here, what are the questions that I am interested in pursuing. Nothing is set in stone yet because it just started. I need to write a bunch of grants. So it depends on what is the money that I'm taking for what I want to do. but there are a bunch of questions that I'm interested on answering. And then a little bit of wrap up what's in the horizon and what are the classes that I'm thinking about teaching. So if you're grad students or if you are willing to thinking about taking classes on the next few years, this is what I'm going to bring. So my background, I have a bachelor in oceanography and a master in physical and chemical oceanography. In Brazil, your bachelor is 5 years. There are no minors or majors, you just take every single class. And I did in a federal university that was, it is the most traditional university in oceanography in Brazil. My master's was a lot of seagoing, so this is me collecting phytoplankton from a net that we actually built, all the ballast and everything, because we didn't have a lot of money. So this was in maybe 2013, 2012. Once I finished my master's program, I came to the University of Miami to do marine biogeochemistry, work with Dennis Hansel. So I worked a lot on the carbon cycle and dissolved organic carbon, marine productivity, also going out to sea a lot. So these are two pictures from the southern, I think this one is the Southern Ocean. This is an iceberg, I think it's too bright to see. And this is, I think, in the South Pacific Ocean. So yeah, I went out to sea collecting a lot of data. And once I started looking at the data and looking at my area, I was doing work in the Northeast Pacific when I wanted some extra data to complement my analysis, and then I came across BGC floats. So BGC floats were deployed there. They were collecting data for about 10 years. They have data every 5 days, and I was blown away. I was like, wow. We have just some bottle data, just some profiles from seasonal cruises, and now we have all of this data every 5 days. I want to use it. So I used the data, and when it was time to look for a job, I looked for the job in the place where they make the floats and the sensors and the technology. So I landed at MBARI. So MBARI is the place where the director of these two very large programs developed chemical sensors and help building and deploying and managing the data from these floats. The PI is Ken Johnson, so he was my boss. I got an institutional fellowship for a postdoc there, and then right after I got a permanent position there. MBARI is different from academia and similar in some ways. So I was a postdoc for about 1.5 years and then a researcher. I worked for these two large programs, GoBGC, that throws the floats all over the world, and then SOCOM program that throws these floats in the Southern Ocean. It's actually how the program started. Because they deployed floats all over the Southern Ocean and the project was a success, then it expanded to the globe. And in this lab, as a researcher, I did a lot of tech development. Again, it's not a university, it's a research institute. So we do a lot of technical work, engineering work, not necessarily science and then papers, but a lot of instrumentation. These floats give data that are then freely available. So this is more or less how we wanted to give the data to the community. So this is the goal of the project. So I did a lot of sensor development, sensor calibration, and then I was exposed a lot with the data, and then I was able to do a lot of science with the data because I knew where the floats were getting deployed, how the data looked like, what was interesting, what was not interesting. I got that firsthand, so that I was exposed to something a little bit different. And then I did a lot of outreach because the data, there is a big portion in those projects about outreach. So adopt a float program, developing ways of communicating what the float does to scientists that are not only interested in the biogeochemistry, but using big data sets. So this is MBARI, just talking a little bit about MBARI. This is San Francisco. It's in Central California, about an hour from San Francisco. It's a research institute that is very engineering based. So they develop platforms, they develop sensors, they develop cameras to advance our understanding of the ocean. So it's very different. It is a non-profit organization, and it's under Packard Foundation. So a lot of our money is hard money and comes from the Packard Foundation. So you don't really need to write grants and proposals. You just get this internal money and do very high risk science. So any science, any development that you want to do that is expensive and that agencies will never give to you, it's possible at MBARI. More recently, MBARI started allowing us to write grants, and this is how GoBGC and SOCOM became part of our lab. But before that, it was only sensor development. And yes, so this is MBARI, it's beautiful. It's in front of the ocean. It's in a 700 people town. It's a little bit far away from wherever you want to live, but a very pretty environment. This is just I put a video here just to show the types of technologies that they develop, and it's very interesting. So a lot of vehicles. So these are the floats, but like benthic stuff, rovers, imaging for deep ocean. Because MBARI is in front of a canyon, they do a lot of deep sea research, a lot of biology, cameras to capture fish and very small plankton, sediment traps, so lasers to measure the sizes of fish without the need to go there and capture them, lots of ROVs and AUVs, gliders, so they develop all of these types of stuff. And we can test right in front of our front yard because the canyon is right there. So this is just they just started with the aerial system imaging, so imaging seagrass and corals and stuff like this. And then something very strong in the institute is, let's see, the animals. So MBARI is also very big on outreach. So all of the figures and pictures of animals that they discover, and they discover many new species every year, go a lot to social media. So they're very big on social media, trying to bring science to the general public. So yeah, they find some very interesting new creatures, and it's very pretty. Even though it's very far from our field and from my field, but it's very pretty and interesting to be exposed to these things. This was my lab, Ken Sensors lab. So it's a giant lab. So we worked with sensor development and then calibration, making these floats over here, then deploying them and working with the data sets and quality controlling. So it's a large group of people. And this is during COVID. And we can decorate our floats, and sometimes we are lucky enough and deploy them. Yeah, I did a lot of sensor production. So these are the heads of the floats. These are all the electronics. So the sensors are basically on the head, most of them, other than the biooptics. So this is part of the nitrate sensor, the spatiotemporal, the lamp. Here is the probe that measures the absorbance of the water to convert into nitrate concentrations. Here is a pH sensor, so we all make those in the lab. And then we send these heads to University of Washington. So University of Washington has the Argo program and the BGC-Argo program, and they put all of this together. They test in a pressure chamber, and then it's ready to go to the ocean. Here is at the University of Washington in the lab, and these are all Argo floats. So they have a very big facility there. So this was basically my life before coming here. And then you have all of these floats that you can work with the data. And these are only the active floats right now, as of December. But you imagine that a lot of them already died. Oh, they last about 10 years. And they last about 5 to 6 years, and then they die, and then they become inactive. So these are the active floats, and you can do a lot of global analysis, or you can use one single float, and it gives you a lot of information about an area. So I'm gonna talk a little bit about these BGC floats and what do they do. So I am interested in areas of the ocean that are under changes. So for example, the biological carbon pump, which is the mechanism that takes carbon from the surface of the ocean and pumps to its interior, and it helps controlling the Earth's climate, is changing right now. And it's supposed to be more weakened in certain areas. And I am interested in those areas. You can estimate biological carbon pump using BGC floats because it gives you very complete data from a particulate organic carbon, which is the carbon that is sinking to the deep ocean. These areas that are decreasing, that this biological carbon pump or BCP is getting weaker, are also under extreme warming events like marine heat waves, like El Nino events. They happen to be in those regions. And then those regions also happen to be oxygen minimum zones. So regions in the ocean where oxygen becomes almost anoxic. And I am also interested in looking at these places. And with all of these floats in there, and the data is freely available, it's very convenient to use them to then understand the changes, the biogeochemical changes in these oceans that are changing. So basically, once these floats are deployed in the ocean, they go down to 1,000 meters, they drift, and when it's time to sample, they can go down to 2,000 meters, and they start ascending. They turn on all of their sensors and they measure the ocean. So all of the sensors measure the ocean at the same time at high vertical frequency, so very high vertical resolution. These are the sensors that can be deployed in the floats. They are very limited by battery power, so it takes a very special sensor to be able to put in a float. And then you can do whatever you want with these sensors, and people have been very creative on using the data from these floats to constrain a lot of different biogeochemical cycles. They are also the dominant source of BGC float data. So this is, for example, nitrate measurements, number of nitrate profiles over time. And nitrate is a very important nutrient for primary production. So ocean productivity is measured by the amount of nitrate that the phytoplankton picks up. And this is shipboard data. This is float data. So as you imagine, if you use these in combination with these, it's very powerful. They are also very accurate. So this is float oxygen, nitrate, and pH from their first profile. And you can match with bottle data. So bottle that scientists collect the sample in the field and measure. And if you do a plot, you can see that they're very reliable. So our data scientists work on quality controlling this data set. There is post-quality control, but they work, it's a lot of work. It's a whole other world. The quality control people do an amazing job and crazy amount of work to make this data reliable enough so the end users can use without a problem. And then you can do all sorts of research. I'm just gonna show 3 examples very quick, but using one float, for example, you can calculate POC fluxes, net primary production, so how much carbon do phytoplankton produce over the year, how much of this is going to sink, what are the fractions of DOC, POC, and PIC that are being exported, all using one float and a little bit of shipboard data just to validate results. So one float already gives all of these things, all of this information about carbon cycle. And this was done in the Northeast Pacific Ocean. With a few floats, we can even take a look at how plankton are placed in the water column. So these are floats that were deployed in the Pacific versus in the Atlantic, in the subtropical zone. And you can see that the placement of these particles are different in between the 2 basins, for example. And large particles were present differently as well. And so using data from these field floats, we could see that large particles, for example, were placed under the mixed layer, while small particles were more spread throughout the water column. And this just tells me that different assemblages of phytoplankton live at different areas in the water column, which was very interesting. And then you can use this to calculate biological carbon pump, transfer efficiencies, and this is what we've done in this paper. And then with many, many, many floats, you can start constraining a lot of stuff. So in this paper, we constrain net primary production. So the amount of oxygen that the phytoplankton produce in the world's oceans using BGC float data. So in the past, this was done using bottle data, and this is how it was done in the past. This is how we did in the present because we have lots of floats. They measure oxygen very reliably, and they are more homogeneously spread throughout the world. And so by doing this, we could estimate net primary production for the globe, but also by latitude. These black dots here are from floats. We also ran 3 models that use satellite data to do net primary production, and then we compared with our float data. And we found out that the global net primary production using floats with our method is very similar, gives very similar values as the ones measured by satellites. So this tells us that it looks good. It looks good. Then now 3 examples of things that I have been working on, just so you have an idea of what I will be bringing in these next few years here, because some things are just done, some things are still under development. So this is a paper that we just finished. It's under review. It's on Nature Communications, and it's the impact of this marine heat wave here in the carbon cycle and the microbial community composition. So there was a marine heat wave in the Northeast Pacific Ocean, and it really disrupted the system. So this one happened between 2013 and 2015. The second one happened between 2019 and 2020. It brought a lot of warmth to the upper ocean, stratified the system, disrupted the carbon cycle, community composition changed. We happen to have float data here, collecting data before, during, and after these marine heat waves. And we were very interested in understanding what happened with particles, what happened with carbon. Do you have more production, less production during the marine heat waves? And if you have a change in production, who are the producers, who are the consumers? Did the food web change? So we use data from floats that were deployed there. We also use data from remote sensing because our chlorophyll needed some corrections. We use satellite data to correct for that. And we use cruise data. So cruise data to collect genetics to pair with the float data to then target, because there is a time series that comes here every season and every year collecting this data and collecting these genetics. We analyze these genetics to then pair with the float data and then see how those changes took place and if those changes took place. So just a very quick results. 2 years of the marine heat waves. Marine heat wave 1, year 2015. Marine heat wave 2, year 2019. We had this anomalous production of POC, and we have anomalous export and retention of POC in the mesopelagic. So during these marine heat waves, basically the POC, instead of being produced, instead of sinking, it was retained in the mesopelagic, which was very weird. So if you think that carbon export, the deeper it goes, the more trapped it gets in the ocean, that was not the case during the marine heat waves. So we targeted these 2 areas, and then we know that this POC export was triggered by the spring blooms, so we already knew what to look for. So this was something related to changes in the food web during springtime. And we know that this region is strongly tied to vertical migration. So there is a lot of zooplankton doing this transfer from POC to the surface to the deep ocean. And in fact, when we go to the genetics, And this is a lot of data, and I don't want you to look at the data. I just want you to see that the green is 2015, that first marine heat wave. 2019 is in orange. This is surface, and this is mesopelagic. A lot of changes were done in the mesopelagic. So these colors show what are the organisms that were present during those marine heat wave years that were not present before. And we see organisms that were never present there, like Acantharia, which is we don't have a lot of information about it, but it was really present during 2019. And it's a big, it's big on particle fluxes and trapping carbon. And we just saw that there were a lot of changes in that mesopelagic area where that PLC was accumulating. So that was very cool to be able to pair the float data to then look at these fluxes and then the genetics to look at what was going on with these particles. Why did we have those anomalies? Now, this is an ongoing paper that I'm working with Kaylee Jones. She's a PhD student at Scripps, working with Lynn Talley and Matt Maslow. She's a physical oceanographer, and she was very interested in looking at the floats that were deployed in the equator. These floats have been measuring biogeochemical variables for the past 4 or 5 years, so very recent. And more recently, we had more data. So we had a lot of data coming up, and we wanted to see the effect of the different ENSO conditions on ocean biogeochemistry. So this was an idea that I took from a paper that I wrote 2 years ago using shipboard data that were reoccupied every 10 years. They're the Gold Ship program. And if you look at nitrate, which is that nutrient that fuel primary production, during different La Nina or neutral years, there is different nutrient concentrations near the equator. So I was thinking if there are differences in nutrients, we might see differences in biogeochemistry, maybe in primary productivity, maybe increases or decreases in chlorophyll, PLC. What are those changes? Now we have a high resolution, high spatiotemporal resolution, and we can solve for that. So Kaylee is doing a very good job. So these are the float data that we have in the region, multiple years of data. We divided these regions in NINO indexes, and then between north, central, and south equator. And then we also use the subtropical zone, which is something that people don't really take a look at when they analyze ENSO conditions. But we thought that, well, a lot of these nutrients are being transported to this transition zone. This transition zone is very susceptible to these changes in the ocean circulation. So what would happen there if there is an El Nino, La Nina, or neutral conditions? So we separated this area. We have tons of data. I'm just going to give you a glance of 2 data sets. So for this region, NINO 4.0, that the physical oceanographers are used to naming, we see these are mean profiles using all of the floats, and then separating these floats into El Nino, La Nina, and neutral conditions, and looking at its average profile. We see that during La Nina conditions, it's colder, and during El Nino conditions, it's it has less salt in the upper ocean, right? So there are these changes that happen between these ENSO conditions. A lot of works have been saying that during El Nino conditions, there is less productivity, but they all use satellite data that only look at surface. What we see with our data is that there is actually, this is chlorophyll. So let's imagine satellites are measuring chlorophyll. Now we have in depth values of chlorophyll. There is actually a displacement of chlorophyll. And just basically the phytoplankton is being displaced a little bit deeper during El Nino conditions. When we calculate stocks of particles and stocks of chlorophyll, we don't really see a big change in productivity, which is also very interesting. And the reason why this chlorophyll is being displaced a little bit deeper is because during El Nino conditions, there is no nutrient in the upper ocean. So this phytoplankton here is being drawn to the deep ocean to grab nutrients. So this is what we are observing. So this is basically this zone here where the phytoplankton is migrating near the mixed layer in these dashed lines over here to grab nutrients. And then when we go to this transition zone over here, there are not a lot of changes in the physics, but basically there are also not a lot of changes in particle production, in chlorophyll production, but it's because this region is never limited by nitrate. So we believe that these changes in ENSO conditions are not bringing anything limiting or something that is going to promote some growth. So it doesn't matter the ENSO condition, this area is not really dictated by these conditions. And it's basically because this area is really limited by iron, so we think that iron is not reaching there, independent of the ENSO condition. So this is an ongoing work. And then I am also using floats to constrain the biogeochemistry of oxygen deficient zones. And if you saw my talk a few weeks ago, I talked about this. So I'm basically using the nitrate sensor to estimate concentrations of other chemicals in the water column. And these chemicals are very important in areas where oxygen reaches zero, because microbes don't have the oxygen that is free for their metabolic incorporation of organic carbon, so they start respiring other components. So they really change the biogeochemistry of the ocean. If we can measure certain concentrations of certain components, then we can start putting things into models, and then we can start understanding where these microbes are transforming these elements and in what regions, in what rates. So this is the idea behind this work in here. And the two chemicals that I can measure, they are part of these microbial transformations. So this is cycle of nitrogen in the ocean, very tied with carbon. So we can look at the float data and tie nitrate sensor with these new chemicals, which is nitrite, that we can now develop. We now develop the method to measure, and then with POC, which is the biooptics from the sensor, to then understand biogeochemistry. This paper is already, it's under review. It's in Global Biogeochemical Cycles. We already have the preprint in there. And basically we have the measurement of nitrite and thiosulfate using the same sensor that was before used only to measure nitrate. So basically I picked the raw output of the data set, reanalyze in a different statistical approach, and then we can get those concentrations. And then you can reanalyze the entire global data set of floats, and in regions where you have these two components, then you can start seeing this very high resolution of data and how they change over time, which was not possible before, because these chemicals are not really measured in very high resolution, only if you go on specific cruises to oxygen deficient zones. So this is going to open a lot of questions for us to answer from this data. We validated our method using bottle data. So we use cruises where they deployed floats and happen to be collecting nitrite data, very good nitrite data. We validated our data set, our methodology. And then you can have this time series of all of these different components, all from float data. So for example, you can see the chlorophyll varying. This is a float deployed in the Eastern Tropical North Pacific ODZ, one of the biggest ODZs in the world. And you can see, for example, how particles behave. So these particles over here, for example, are below the euphotic zone, and they are from bacteria that are doing all of these transformations. And if you pair this data with nitrite data, thiosulfate data, this new approach, you can see that they match with the regions where these chemicals match with regions where these bacteria are in very high concentrations. So these areas are areas where oxygen goes near zero, and you can see from the oxygen sensor over here, all that is white is where oxygen is almost zero. And you can see also from this data set, all of these blips over here, these are submesoscale features. So this area is very active on eddies, little eddies that pass, And then you can see how this oxygen is getting injected inside the ODZ. When the oxygen gets injected, then bacteria are like, yes, oxygen. So they stop, using nitrite and thiosulfate, and they disappear from the water. So it's very cool to see all of these biogeochemical transformations happening using multiple sensors that are independent from each other. And I know it's a lot of information, but this is just showcasing how we can use everything together, and then it gets you a complete story of biogeochemistry. So my PhD student that is coming this fall and hopefully already accepted my offer, he's going to the Bay of Bengal with me. This cruise is going to happen between June and August. We will keep working on the oxygen deficient zones. Bay of Bengal also has an oxygen deficient zone, and we are going to be measuring more nitrite, more thiosulfate, and then deploying a nitrate sensor on the rosette to help validate even more this methodology. Go BGC is just about to deploy a couple more floats in there, so my PhD student is going to be working with that data set, with the cruise data set. I have a cruise in the Santa Barbara Basin where I'm going with MBARI. Santa Barbara Basin is another anoxic environment, and we are going to do some more research on ODZ and with sensors. We just want to strengthen this method. Once we have this method strong enough, which we already know, or we already think it's strong enough, it's validated, but once we have a little bit more, even more data, then we are going to do the global nitrite and thiosulfate data set. So we're going to reanalyze all of this float data and give all the new, we're going to have all the concentrations of nitrite and thiosulfate distributions in the globe, and this is going to be part of my PhD student. There is a lot with method and sensor development that can be going on. MBARI is still a partnership. I am collaborator at MBARI, so we will still be working on sensor development and methodology development just to give us new tools and new opportunities for looking at ocean biogeochemistry. So I have a lot of ideas for future projects. A lot to do with nitrite data set, because now this data set is so new. The resolution is different. There are a lot of things that can be done. There is an ongoing conversation with a friend to tie production of N2O, which is a greenhouse gas that is produced in oxygen deficient zones, with BGC floats. And then looking at microbial processes. So what microbial processes produce N2O and where and at what time of the year. So this is something that we are going to be exploring. Then interpret the global data set of nitrite and thiosulfate, because once we have all of these concentrations, there's a lot that can be done. ODZs are all different from each other, which is very interesting. So interpreting this data set is going to be, we are going to have multiple opportunities to interpret this. I am also interested on the underuse of some BGC floats. For example, floats that are deployed in the equatorial Atlantic, near the coast of Africa, that are subject to Sahara dust. There is very little that was done. So I'll be very interested in coupling atmospheric data with BGC float data to look at how this Sahara dust, for example, induced net primary production and what's the impact on carbon fluxes there. Impact of ENSO on biogeochemistry, the ongoing work with Kaylee. It's going to be one chapter of her thesis, but I would like to keep exploring this ENSO, especially because we deployed even more floats there. And actually, those floats have been measuring a lot of nitrite, and which is very interesting too. Then tying genetics with BGC-Argo floats. So we saw the power of tying microbial composition with carbon fluxes. I would like to keep doing this, so I will probably continue to be going on cruises to measure things, because to be able to answer some questions, we still need to go on cruises and measure things chemically and biologically. Long term, my lab has space for sensor development and methodology development. My prospective student is interested in sensor development, so we might continue to be exploring that path. There is a lot of, with these new technologies to pair BGC float data with mooring data, time series, gliders. Gliders can hold the same chemical sensors, but they look at the coast where the floats can't go because they bump on the bottom and they stay there and they get stuck. So this continuous between land and open ocean is very interesting and very underexplored. So if we are able to use the same sensors, collect the same amount of data, and then connect things, very interesting for biogeochemistry. And it's already in discussion to explore ODZs using floats that we can purchase and customize their missions. So missions that are targeted to the center of ODZs. So there is a discussion going on already. I was in a meeting this week, and there are some people interested on just writing large grants, because GoBGC and SOCOM are global, and they have these missions that are set up and can't be changed, because the idea of the program is to have an homogeneous type of data set for the community to use, which means that you can't really ask my boss, hey, can you change the mission of a float sample every day in this area that I'm interested? He's gonna say, no, we can't, because it's for a number of reasons. So if we can purchase floats and customize missions, which is very hard, but it's possible, we can then really change we can open other questions. And then here, I am going to be responsible for the marine biogeochemistry class, undergrad and grad students. And I am preparing a new course, which is called Data Science in Marine Systems, which is going to teach grad students and undergrad students on how to use multi-platform data to look at the ocean. So this is ongoing, probably gonna happen next year sometime. I'm working on the curriculum right now. And the idea of the second class is to be a very hands-on, project-based type of class where students can come to the class, learn where the data sets are, what are the types of data sets. You can pick a region of the ocean where you will need to stick for the entire semester, and you will learn everything that is happening in that region and what types of data can you access to interpret the data set, to interpret the system. So this is the idea. It's not going to be biogeochemistry based, but it will be encouraged to be biogeochemistry based. It will depend on your interest. And yes, the idea is to use remote sensing, shipboard data, moorings, Argo, BGC-Argo data. So it's gonna be a mix of lectures and a mix of hands-on project. And I hope to have that available soon for our students, because it's something that I missed when I was in grad school. And when I was in undergrad school, I learned by trying. So I hope these will help people and open possibilities for people to look at different types of data. And then with that said, thank you. This is all I have. It's a little glossed over, but yeah.
Yeah, thank you very much. I think Igor is still in his meeting, so I will moderate the questions. Do we have questions from the audience here? No questions?
I have a question.
Yeah,
but
Lisa.
maybe I should let the young ones go first.
Maybe you will encourage them
You
to
are
ask
the only
questions.
one.
So Mari, you mentioned how exciting. So glad you're here. You mentioned the calibrations or the checking of the floats with the bottle data, which is brilliant, right? How do they get to instrument drift, sensor drift?
They get the drift so the data matchups bottle, they are not made for correcting sensor data. They are just a validation. The corrections are made using algorithms and glow depth data. There is Canyon neural network. So basically, you match the sensor profile with profiles that are made synthetically from these neural networks and glow depth data and stuff like this, and then you compare them, and then you assume that the deep ocean is not changing by much, so that this location of the float is the drift.
Okay.
And it's a little bit different from sensor to sensor. For oxygen is a little bit more complicated, for pH is a little bit more complicated, for nitrate is easier.
So it sounds like kind of similar techniques to the physical Argo program.
Very similar.
And you know, one of the issues with that is we know even the deep ocean's changing now.
Yes, but we try to use the data that is as recent as possible. I know that the neural network has been using very recent data, like Canyon B and Asper, which is two data products. Yes.
So those Gosha crews are still super
extremely important. Without the cruises, we would not be able to use BGC floats. Yeah, absolutely not.
More questions?
Yes.
Sharon.
In the Eastern Atlantic, there's been some pretty substantial warming just sort of just west of the coast of Africa, and I look at it just for hurricane research. Have you or anyone looked at that region that you mentioned about the Saharan dust influence, which could be significant? Is there also, have you seen, perhaps due to a lack of upwelling, just sort of changes in the sort of biological and chemical distributions there? So do you have an idea?
There are a few people working on the North Atlantic, more like subpolar area. And there are a few papers that have been coming out in the past 1 or 2 years in that region, but nobody has been looking at the tropical area, which is very interesting. Yeah. We should look. There has been some work on like near here, looking at hurricanes, so impact of hurricanes. Jen McWhorter just published a nice paper on that. So yeah, but that area like near Africa, not a lot of things at all. Yeah.
I'll just say one thing about that, because I have a postdoc from West Africa, and the piracy issues are keeping oceanographers out of that area, so it would be really great to look at float data there.
Yes, we deployed some floats in West Africa. In that area, very west South Atlantic Ocean, in the belly, we have a bunch of floats there, and it's a suboxic area, and it was a big gap in data there, and we went to a cruise last year, deployed a bunch, like 6 or 8 floats. So we have a bunch of data right now, less than a year of data, but a lot of data. So yeah, there is a lot. It's so many floats you can't really keep up with, you know. And a lot of opportunities, especially once they collect data for like a year or so, then you can have like a variability, right?
Any more questions from here? I don't see any questions from the Zoom audience. John, I muted you. If you unmute yourself, I think you can speak to us.
Oh, muted.
You're muted.
Okay.
Now.
Okay, can you hear me now?
Yes.
Very nice talk. I'm sorry I didn't get a chance to attend in person. I've got workmen in my house today working on a high efficiency air conditioning system. But I developed the CycleSonde, which is the great grandfather of your profiling technology. And maybe I'll give a talk on it, but I would, have you considered putting your suite on profilers that are moored?
To put the sensors, the same sensors on moored?
Yeah, moored profilers.
Yes,
Yeah.
Yes, we thought about it. Well, you can't do everything, right? But it will be very
no, you
interesting.
can't.
It will be very interesting. Actually, at
Yeah.
MBARI, our mechanical engineer developed a coastal profiling float. And it uses all of the sensors that we have on the BGC float, and we deploy in the coast, and then everything is so strong and dynamic that we always lose the float. So he's like, we should use the coastal profiling float, the CPF, which I called Frankenstein because it's very ugly and he doesn't like it. But we should do it moored, because then we wouldn't lose it. But there are ideas. There is just nobody executing them.
Yeah, one of the interesting byproducts of profilers is you can have them inductively coupled, so you can get real-time data at high resolution.
Yes. The CPF was profiling every like 4 hours, 3 hours. We have a postdoc that is brilliant, Magdalena, and she was trying to get measurements of vertical mixing using that CPF. So profiling at very high frequency, very high resolution.
Yeah. Well, very interesting. I look forward to meeting you in person and having a longer chat with you.
Yes, absolutely. Come by my office.
Okay, will do.
Someone typed a question here. Would you be able to unmute yourself and ask that question through the microphone, or should I read it? Amber?
Hi.
Yes.
I was just wondering if you could expand on like your use of genetics and how that kind of intersects with the chemical data you're getting.
Yes. So we were lucky enough that these floats have been deployed in an area that doesn't move a lot. So these floats were hanging out in the station P, which is the last station from the line P, station Papa, the last station of the line P program. It's a Canadian program from Fisheries and Sciences that goes there for the past, I don't know how many decades, and they collect a lot of chemical and biological data. They happen to be collecting genetics for a while, many years. They were just targeting heterotrophic bacteria because they were looking at the changes in heterotrophic bacteria. So my last chapter of my PhD, I published a paper looking at the impact of that first marine heat wave using BGC floats, and I happened to give an interview to a journal, and the person that did the genetics for the heterotrophs was interviewing for the same article. So we met, and I told her, I have a lot of POC data that you said in your paper that you're lacking to look at the BCP, biological carbon compound. Do you want to collaborate? And she's like, yes, let's reanalyze our samples for genetics. So we reanalyzed their samples for genetics. It took a very long time because they were all frozen on the back of the freezer because nobody was using it. So we did metagenomics, and we targeted everything from eukaryotes to zooplankton, protozooplankton, microzooplankton, everything. And we targeted 2 depths, the euphotic zone and the mesopelagic. And then for multiple years. So we had years before, during, and after the marine heat waves until 2021 or 2021 because the COVID hit and they stopped collecting it. So this was not a funded proposal. It was just curiosity from a couple of scientists, Stephen Hallam from UBC, Colin Kellogg from Hakai University, Angelica Peña from Fisheries in Oceans and Fisheries, and me, and yes, and a little bit other people. But yes, it was metagenomics. That's the answer.
Okay, I think that's the end of our seminar. Thank you again. thank you all for coming. We will now take a little break with the seminars. The coming 2 Fridays, we have some other activities here. And next Wednesday, there's no seminar for unknown reasons. And then in 2 weeks, we will have a visitor, an interesting visitor from Japan, I think. And then the first student seminars. So come again in 2 weeks.